about beta.
Okay. Good evening, Pratham. Okay, so I'm Rahul Saha. So today we'll be discussing about the types of conduction, the neurotransmitter release, and also the types of neurotransmitters if we have enough time. Okay then. So in the last class we had discussed about how does the you can say resting membrane potential stay to be maintained, or how does it stay maintained, and how does it occur, how does it perpetuate, and how does the current propagate from one specific point to another specific point. Okay, that is what we had talked about in the last class. In today's class we'll be talking about the specificities of myelinated axon and non-myelinated axon. Okay, good evening, Gayatri. So we'll be talking about how does myelination play a great role in axon and in the current propagation in axon. That myelination is not just for insulation, that is correct, but it also plays a drastic role in the propagation of the current. Okay, that I'll show you. And one more thing I'll tell you is, after the action potential is over, how do we rebalance the entire. Okay, doesn't matter, Gayatri, you can sleep. No problem. We'll do it again. Okay. So next class we'll do it on Zoom. Okay, no problem. You can ask your queries, clarifications. Okay. Okay. So the thing is, next we have. Just open. Okay. So what I want you to know is, see, after the action potential generation, right? After we have reached minus 70, then suppose minus 55, that is the gradient membrane potential, then you have 0, then you have reached plus 30, plus 10, okay? So you have reached like this, then you have been able to come little bit down, you can say minus 90 till here, okay? So this is the time period, x, y, 0, 0. This is the millivolt potential difference, right? Now the thing is, here the potassium channels will be inactivated. This is your depolarization. This is your repolarization. Now just tell me, here Na plus influx is happening. Okay. Here K plus efflux is happening. Okay. But one more thing I'll tell you is, in a specific active neuron or in our neurons, what happens is, this current gets propagated, or this action potential is generated almost 1000 times per second. Okay, that's the enormous amount of time the action potential is generated in our neurons. Now, after it has been generated 1000 times in a second, what do you think the consequence be? What do you think the consequence will be? You can say, I had told you that the Na plus concentration is always more extracellularly compared to its own concentration intercellularly, and the K plus concentration is always less extracellularly compared to its own concentration intracellularly. But when the influx of Na plus is occurring continuously, ceaselessly, incessant efflux you can say, and the K plus efflux occurs again ceaselessly, won't the concentration try to flip otherwise, right? If we don't keep a check, will the concentration not happen like this or not become other way around? Will the concentration not become like this? Na plus almost increased, concentration of Na plus decreased extracellularly, increased intracellularly, and K plus decreased intracellularly and increased extracellularly as a consequence of this very influx and efflux, right? This is influx, this is efflux. So this will be happening or not after a certain point in time, that you keep on going for the action potential generation. Every cycle you are depolarizing, again you are throwing out K plus, so it gets repolarized again, minus 70 is reached, again depolarizing, again Na plus comes down its concentration gradient. So after a point in time, the concentration gradient which it is following Na plus may not be present at all, right? It may not be present. So what will happen there is, you will have a specific problem here. A specific problem means you will have Na plus level rising and K plus level decreasing in the cell, and the problem then that will pose is, now in depolarization or under the phase of depolarization, will Na plus try to come? No, it cannot come because the concentration gradient has reversed. Same way, K plus cannot be efflux, again concentration gradient has reversed. Okay, so this entire thing will be reversed then at, you can say, minus 55. Even if voltage gated, you can say Na plus channels open up, still Na plus cannot come in, right? Here what happens at minus 55 voltage gated, Na plus ion channels, they open. Even if they open, if the concentration gradient has been reversed, how will it come? It is not an active transport, it cannot come in. So what will happen is, now the current cannot be propagated, nor neither can be generated in a neuron, thereby we will undergo paralysis. The effect will be paralysis. The effect will be sudden death. We cannot even think. Nothing can happen. The effect will be death. Now, how does the body circumvent this problem? How does the body find remedy? Again, our body is very, very clever, very, very amazing. It is full of astonishing, you can say, entities, surprises, you can say. Now, what does it do is, I had told you, if you remember, in one of the Zoom classes only, that our body, our cells, they do contain something known as Na plus K plus ATPase. Okay, Na plus K plus ATPase is a P-type ATPase which is meant for active transport, because Na plus concentration is more extracellularly compared to its own concentration intracellularly. K plus is more intracellularly compared to its own concentration extracellularly. But what it does is, Na plus K plus ATPase, it will break down one ATP molecule, adenosine triphosphate, gamma phosphate bond will be broken down, ADP will be produced, and one pi phosphate will be produced, and energy will be emanated. Now this energy will be used up in order to throw, okay, in order to throw 3 Na plus against its concentration gradient. You can see still it is more outside, but from inside more and more Na plus is being thrown out of the cell against the concentration gradient, and 2K plus is thrown inside. Okay, against again the concentration gradient of K plus, because K plus is supposed to go out but it comes in, Na plus is supposed to get into the cell but it comes out of the cell. So how is it able to go against the concentration gradient? Is due to the energy that has been derived after the hydrolysis of an ATP molecule. Thus, this is known as a completely active transport. Active means using the energy derived from ATP hydrolysis to cause transport of ions against their concentration gradient. So this is the nature's answer. So once the concentration gradient becomes quite lesser and lesser, this ATPase, Na plus K plus ATPase, it opens after this. At this stage, it will open up and then it will reverse the whatever concentration gradient loss that has happened due to Na plus influx and K plus efflux. Again, setting up the concentration so that the next action potential, okay, can be triggered properly. So it won't happen after every action potential. After 10 to 15 action potentials, when the concentration gradient really it starts to diminish, then it will commence and it will again reset the concentration gradient so that Na plus is less inside the cell, and at minus 55 millivolt of the voltage, Na plus voltage gradient channel opens and then Na plus can come down its concentration gradient again. Okay. And one more application of this specific channel is its electronegativity. It is causing electronegativity how? If you just check the number of ions being thrown in and thrown out, okay, 3 Na plus means 3 positive ions are thrown out, 2 positive ions are thrown in. So after every round of ATP hydrolysis, what it is doing is it not causing one positive ion deficiency inside the cell? Because it is throwing out 3 positive, only bringing in 2 positive. So it is causing every time one positive ion or one positive charge deficiency. So this is also the reason the electrogenic, it is known as electrogenic nature of Na plus K plus ATPase is also one of the prime reasons behind the resting membrane potential being negative, that is minus 70 millivolt, because it is causing the negativity inside the cell to be promoted, right, by a plus 1 charge every time. It is causing deficiency of plus 1 means it is causing an excess of minus 1 automatically if you see from that perspective. Okay, so that is, now we have completed the action potential completely. Okay, now we have completed the entire action potential. How will the ion balance be restored also via Na plus K plus ATPase? We have completed. Okay, so this is how action potential is completely understood, and it is completely supposed to be learned via conceptual understanding. Now, coming on to myelination and non-myelination. Okay, so there are two types of conductions, okay, that is affected by myelination and non-myelination. Okay, so the thing is, suppose I'm drawing only the fiber, the axon. Okay, the axon here is myelinated. Okay, so I have told you all the axons are not myelinated. Remember that. Okay, so here suppose this axon is myelinated. Next, this axon is not myelinated. It is an unmyelinated. This one is myelinated. Axon. This one is an unmyelinated axon. Now, what would happen in these two scenarios? Scenario number one, scenario number two, with regards to electric current or action potential propagation. What I say is action potential propagation will be much faster in the myelinated axon, much faster in the myelinated axon compared to the unmyelinated axon. Now how? Let's check this myelin sheath. So myelin sheath is made up of myelin basic protein, okay, and mostly the lipids. Now, what do you like to tell me about the lipids? The lipids are non-polar. They won't allow any kind of conduction, right? Will they allow any kind of electrical conduction? No. So whatever conduction has been generated here, okay, so a local current will directly pass from here to here, from internode to internode, come to the node of Ranvier, okay, it will come to the node of Ranvier and be again a new action potential will be initiated at the node of Ranvier, then again local current, it will influx, it will be again, it will entirely skip, it will cause a jump, okay, it will cause a jump from one node to another node, okay, sorry, from one internode to another internode it will jump, okay. Only at nodes which is known as the node of Ranvier will the action potential be again restarted or recommenced or reinitiated. Again, one more concept is it doesn't have to go through the entire membrane. Just like I showed you in the last class, that the action potential is being propagated from one region of the membrane to the other, from one region to the another. You can say millimeter by millimeter it is propagating. Meaning what? Everywhere or at every locus of that membrane is the action potential being again recommenced, reinitialized, terminating Na plus K plus pump after few action potentials are again starting. Again it is being recommencing, again depolarization, hyperpolarization, again depolarization, repolarization, hyperpolarization, again re hyper. This continuously, ceaselessly is going on every millimeter of the locus pertaining to the entire axon. Okay, entire you can say axolemma, right? So that is what will happen in this case, unmyelinated. Here plus, okay. So some plus will jump from here to here, okay. Then this will become negative, this will become plus, okay. Again it will jump from here to this will become plus, this will become negative now because of repolarization, and every it will go through the entire, or you can say it will go through the entirety of the plasma membrane, okay, not leaving even a nanometer. Action potential will be regenerated again and again and again and again through the entire plasma membrane. But in the case of, you can say, this thing, the local current will not go through. The local current will be produced or will produce a gradient potential, okay, that brings the axon membrane or axolemma at the next node to threshold. That will it do, and then action potential will be commenced again, okay. So this is what will happen always, okay, in a myelinated axon, okay. So action potential will develop at node 1, then at node 2, then at node 3, okay. That is how it will keep on happening, okay, local current only. But in case of unmyelinated axon, it will go through the entirety. That is the concept here. It will go through the entirety because there is no non-conductance. Because in case of, you can say, the presence of myelin acts as an, you can say, impervious resistance, a very, very high resistance which cannot be overcome. So it will jump from node to node, okay. It will jump from one internode to another internode. So node to node jumping happens because the resistance is overwhelming. But here resistance can be overcome. The resistance is being provided by the plasma membrane also, but it can be overcome. So it is being overcome. Okay. And then action potential is repeatedly being reinitiated, and again it is propagating through every nanometer present in the cell. Okay. So this type of action potential, okay, there is a name also, I'll tell you the name. This type of action potential or this type of conduction is known as discontinuous conduction. Okay, discontinuous conduction, because you can see it is generating at node to node via local current diffusion. Okay. Also there is a specific name in case of NEET examination, if you can note it down, it is also known as saltatory conduction. And this one, since it is going through the membrane nanometer by nanometer, it is known as continuous conduction. Okay. So the same concept of action potential propagation, just for the NEET examination, the name changes as saltatory or discontinuous. You can call it discontinuous also, but in NEET question they'll ask about the saltatory conduction directly depending on the myelin sheath presence and non-presence or absence, you can say, right? So this is your complete evaluation of the conduction in the myelin sheath. Now, let me tell you about the three types of axons also that I was talking about earlier. The three types of axons that I was talking about, and how does the axon diameter, because we had talked about that equation R equal to rho L by A, where I told you the axon length, if more length is there, the resistance will be increased because the numerator is being increased, and if more diameter is there, the denominator is being increased, more surface area you are providing, so resistance will become lesser and the propagation of the current will eventually be faster, right? So like that, we have three types of axons which are asked in the, especially in the NEET examination. So I'll just tell you in very briefly about them. So we have type A fibers, type A fibers, we have type B fibers, okay, and we have type C fibers. So let's first write about or know about the type A fibers only. So type A fibers, once we talk about, okay. So type A fibers, always know that these are the largest diameter myelinated axons with a diameter of 4 to 20 micrometer, okay, diameter of 4 to 20 micrometer, largest diameter. Number 1 point, myelinated axon. Number 2, they are all myelinated axons, okay. Now what is the speed? I'll just tell you the speed, you don't need to remember it, I'm just telling you for your knowledge only. So these fibers can carry action potential at a speed up to 120 meter per second, okay. Speed of action potential traversal, action potential conduction, you can say, that is 120 meter per second. Very clearly know it, okay. Then we will compare one thing which will make you amaze even more. Now type B fibers. What are type B fibers? They are also myelinated, okay, but diameter is smaller than type A, okay. Myelinated axon diameter is 2 to 4 micrometer only, okay. No need to remember the diameter also, just I'm telling for a perspective so that we can compare, okay. So myelination is there, so myelinated, only you need to know which are myelinated, okay. That's all is asked in comparative exam, that type A and B are myelinated, type C is not myelinated. Nothing else will come. I'm just explaining for giving you more of concept that you should know what am trying to convey. Okay. And speed of action potential. Conduction is almost equal to, you can say, 18 meter per second. Okay. 18 meter per second. I'm just explaining that, giving you the speed value so that I can relate it to what I told regarding the myelination. Okay, how does myelination play a huge role in the increase of the action potential traversal or conduction with regards to diameter. The diameter is fine. If you increase diameter, it will increase the speed of the propagation of the action potential, but not so much so with the myelination or unmyelination criteria. Okay, so type C fiber, they are the only fibers which are unmyelinated and least diameter. So diameter is the least, 2 micrometer, unmyelinated. Okay, very important for comparative exam. They are unmyelinated axons. Okay. Now speed of action potential conduction. Speed of action potential is only 1 meter per second. Okay. So now get the perspective. Okay. Now the advantage of myelin becomes clear when you do compare type C to type A fibers. Just compare these two, type A and type C fiber. Type A fibers are suppose 20 micrometer in diameter. Okay. But type C are 2 micrometer in diameter. So what is the difference? 10 times is the difference, right? 10 times is the difference between their diameter. So I can say that type A 20 micrometer, type C 2 micrometer, so 10 times is the diameter difference. So maximally I can, you can say, increase the conduction velocity 10, or if I am more lenient, I can increase it by the fold of 20, right? So what is the conduction velocity of type C? 1 meter per second. If I increase it by 20, okay, so I'll reach the velocity of 20 meter per second, okay, but the velocity of type A is not 20 meter per second, it is 120 meter per second, means the difference is of 120 times. So diameter is playing a very negligible role here. Myelination is playing the, you can say, the majority of the role, because diameter is only 10 times smaller. 2 micrometer for, I'll write it here, type C diameter. Okay, that is why I wrote the diameter also, to put into perspective why myelination is so much significant. Okay, 2 micrometer, this is 20 micrometer. 4 to 20, take it as 20. What is the difference? Ratio is 1 to 10. So here speed is 1 meter per second. As per that, here speed should be 10. Let's make it 20. So 20 meter per second. But the real speed is 120 meter per second. 120 times. Okay. 120 times is the difference. Why? Because it is unmyelinated and this is myelinated. This is unmyelinated. So myelination plays that great of a role because you are able to skip so much of the length of the axon in between every internode. So there are multiple internodes, so you're skipping almost 90% of the axon membrane or axolemma. So only initiating current, graded potential, then action potential at only the nodes or node of Ranvier. So that is how it will pose a great difference. Okay, that is the reason, because you're able to skip 90% of the axolemma. Okay, so it is posing great difference when it comes to the propagation of the action potential through the, you can say, axon. Okay, in case of myelinated axons. Okay, so is myelination completely clear now? Everything regarding the myelination? Is it clear, completely clear regarding myelination? Unmyelination, I think it's must be clear till now. Myelination and unmyelination criteria. Any doubts in myelination? So next up we'll be talking about these synapses. Okay, very important, synapses, right? So okay, good, right. So let's talk about synapses. So now the most important thing regarding the axon, okay, or the synapses or the neuron, I should be saying, in totality is synapse. Okay. Now synapse are what? There are two types of synapses you should be knowing. First of all, synapse is only not one type. Synapses are of two types. Number one is electrical synapse. Okay. Number two is chemical synapse. Always remember. Now electrical synapse. Okay. What happens is these are the direct connections. Okay. Means the cells are in direct physical contact, you can say. and the presynaptic and postsynaptic neurons membranes of the two communicating cells are locked together at specific or by specific proteins, okay, which are known as gap junction proteins, okay, and that is known as chemical sorry electrical synapse, meaning that electrical synapses are extremely rare in both CNS and PNS. They occur only in some areas of the brain, including the, you can say, vestibular nuclei, which is responsible for balance, ear and all. Vestibular nuclei, the eye, and you can say some ganglia, you can say ciliary ganglia and all. They are present there. Okay. So what happens is, I'll just show it to you. Electrical synapse. Suppose this is one cell, okay, this is another cell. They will be very, very close. The gap will be only of 2 nanometer, and also the proteins they will be having different proteins, okay. These are known as gap junction proteins, okay. So these proteins are known as connexin, okay, and they form a channel. So suppose this is a channel through which the transmitters can travel from one cell to another directly, okay. So this channel is known as a connexon, okay. So this is known as an electrical synapse. This is also present. That's why I'm telling you about this, because this is also present. Don't think that only chemical synapse is present. Only thing is the difference is only 2 nanometer, very, very negligible difference. Okay. So now we come to the chemical synapse, which is there for our most of the understanding. Okay. Chemical synapse. Chemical synapse means both the cells, the presynaptic and postsynaptic. Okay. So the synaptic end bulbs are not in direct contact with the dendritic spines. Okay, there is a little bit of gap. Okay, there is a little bit of gap. They are not in direct contact at all, not via gap junction at all, or not by any other junction, not by any other protein. The two cells are not held. There were difference in electrical synapse like this, but they were held by, you can say, gap junction proteins, but they are not held by any proteins. Okay. So there is a clear descending gap between the two cells, so not in direct contact whatsoever. Number one. Then what we have is, it is produced between an axon terminal, also known as the telodendria or the synaptic end bulb. In some neurons, it is also known as varicosity of the presynaptic neuron. Okay, so it is produced between the axon terminal, also known as telodendria, and synaptic end bulb. In some neurons, it is also known as varicosity, okay, of the presynaptic neuron, presynaptic neuron, and the, you can say, dendritic spine of the postsynaptic neuron. So this between the presynaptic means first one, the synaptic end bulbs are the transmitters, and the dendritic spine is the receiver. That we all know, the dendrites are the receivers of the postsynaptic, means the next neuron in the connection. The synapse is produced. Okay. Now, what is the role of the synapse? The first thing is, what is the role of the synapse, and why is it present? What would happen if it is not present? Okay, so those things we'll be considering here. So number one thing is the role of the synapse that we are talking about. Okay, so the role of the synapse is very, very important. Okay, the main role of the synapse is what? Integration of the nerve impulses. If you just imagine in evolution all the cells were connected, okay, like this. Why do you think the cells are left unconnected here? Okay, why do you think the cells are left unconnected? Because of integration of the impulses, so that one neuron can go for multiple synapsing. Okay, one neuron can go for multiple synapsing, so that it can get these signals from various neurons. And not only is a synapse, you can say, axodendritic, okay, it could be axonic, it could be axosomatic also, means axon to the next axon, axon to the cell body directly, or axon to the dendrite. Okay, so it could be, you can say, I write it here, axodendritic, which we saw. It could be axosomatic, soma or cell body. Then it could be axonic. There could be three types of chemical synapses. Okay, three types of chemical synapses which could be present, and they play a great role. Okay, they play a great role in the, you can say, propagation, or the integration, I should be saying, of multiple neurons, or the multiple presynaptic neurons, action potential driven release of neurotransmitter, and that acceptance by the postsynaptic neuron. Therefore, we can recall a lot. Like I told you about the basal ganglia, it had the claustrum, right? So it can recall everything. How? Because from multiple areas of my brain, the synapses are converging together, and more the width of the synapse, that's why there is a theory that more the width of the synapse means more it can communicate with more of the dendritic spines of multiple neurons, more will be our intelligence or recalling capacity. So we should be learning more and more intriguingly, with inquisitiveness, with curiosity, then only with passion, zeal, okay, fervor, then only we can widen the synaptic, you can say, width, you can say. The synaptic width can be widened also, and we can also enhance our own, you can say, intelligence, because every one of us contains 86 billion neurons. But the thing is, the synaptic connections, the synaptic width, the entire width of the synaptic cleft is not the same in everyone. It is very less in case of people who don't study whatsoever, and people who are real gems like Albert Einstein, Sir Stephen Hawking. Their synaptic cleft was much more, and compared to other anatomical differences, also the sulci were more deep, fissures were deeper, wider. All that happens with extreme practice and some kind of genetic causes also, but with practice also, right? So an average, you can say, synaptic cleft average is 20 nm. Okay, not 2 nm, 20 nm in width. Okay, so that 20 nm is the width of the average synaptic cleft. Okay, 20 nm is the average width of the synaptic cleft. Okay, so that is the average width of the synapse. Okay, and what happens is, what we have is, this is for the cholinergic neurons, means they which will release the acetylcholine as the special molecule, okay, as the special neurotransmitter. Now, first of all, a synapse of a neuron, okay, happens between what? It can happen between a neuron and a muscle, that I told you. It could happen between a neuron and a neuron, and it could happen between a neuron and a gland also. So it could happen between two neurons, presynaptic and postsynaptic neurons. It could happen between a neuron and a muscle, which I told you properly in the locomotion and movement chapter. How does acetylcholine cause the influx of sodium ions? okay, into the muscle cell, causing the depolarization to happen. That is known as a neuromuscular junction. Then it can happen between a neuron and a gland, any endocrine gland which will make it secrete hormones. That is known as neuroglandular junction. Neuroglandular junction. So these are the kinds of, you can say, synapsing that can occur, okay, in case of the synapses. Now one thing I'll tell you is, how is a synapse, or how is a neurotransmitter released from a synapse? Okay, there are multiple proteins. We will not go into that detail. There are snare proteins, V snare, T snare. Okay, then there are synaptobrevin, syntaxin. So many types of proteins are there. Okay, neuroligin, which will hold the entire synaptic cleft together. So we will not go into that detail. We will simply explain why the action potential perspective is also given in your NCERT book, that how does it happen. Okay, so first of all, what will happen is, I'll write it point wise. Make a note. It is very important. That heading could be neurotransmitter release from synaptic end bulb. From synaptic end bulb. How does it happen? Okay, so first thing is point number one. If you see, an action potential will arrive at the presynaptic axon terminal. Okay, because it is arriving, so node to node, even if it is myelinated, so node to node, it will in the end arrive at the axon terminal or the telodendria. Okay, and it will depolarize the membrane of the axon terminal or synaptic end bulb. That is clear. So the action potential, point number one, arrives at the synaptic end bulb, or you can say axon terminal. Now when it does arrive, what will happen? It will cause depolarization. Na plus channels will open, voltage gated, and depolarization will happen. It will cause depolarization of the axon terminal. That is point number one. You can note it down point wise. In NCERT, it is not given point wise. Very layman terms, or very haphazardly it is given. So I have arranged it point wise for you. Now, what will happen now is, due to this depolarization, okay, now what will happen? Understand, the extracellular calcium ions, means the calcium ions which are present extracellularly out of the cell, outside the cell, will enter the axon terminal. Okay, and what will happen then? It will trigger the exocytosis of, you can say, mainly these neurotransmitter containing synaptic vesicles. So number two is extracellular. This depolarization will trigger the extracellular calcium ions to enter the axon terminal. The axon terminal, and it will cause exocytosis of neurotransmitter containing vesicles. containing vesicles. Okay, meaning that what would happen is, I'll explain this part to you. When the depolarization, okay, of the axon terminal happens, okay, the depolarization of the if you can say axon terminal, it will briefly open. Okay, what it will do is, it will briefly open the voltage gated calcium ion channels also in the axon terminal. But you will ask me a question, why not it opened voltage gated calcium channel in the axon only? Because those voltage gated calcium channels, those specific membrane proteins are not present in the initial part of the axon. It is only present at the axon terminal. Okay, that is the reason. So since they are absent, so they cannot produce. Just like in the cyton, the voltage gated sodium ion channels are not present. So current cannot initiate or get initiated in the cyton or the cell body. Okay, but in the axon terminal, you have the voltage gated calcium channels which are present. So they'll open, and then calcium ions will rush into the cell, and that will lead to the formation of various signaling pathways. Okay, suppose depolarization happened, it reached plus 30 at plus 30 millivolt voltage gated calcium plus 2 channel. Ion gated ion channel opens, calcium comes in. Due to the incoming of calcium, different, you can say, signaling pathways CAMP, IP3 and that which is not in your course, it gets initiated, and then what will happen is, a neurotransmitter is packaged into the vesicle. That neurotransmitter could be the most famous one. Let's take the example of ACH or acetylcholine. This is the most famous neurotransmitter. Okay. The neurons which release acetylcholine as neurotransmitters, they are known as cholinergic neurons. Okay, from cholines, cholinergic. Okay, from this only, choline means acetylcholine. Then what will happen is, now acetylcholine has been packaged into the vesicles. Now what will happen is, now acetylcholine, okay, will now bind, acetylcholine will now bind to the, you can say, the cell membrane. Okay, it will fuse with the cell membrane, and acetylcholine will be released onto the, you can say, will be released onto the, this will fuse. Okay, this will fuse like this, the membranes will fuse, and acetylcholine will be released onto the synaptic cleft, will be released onto the synaptic cleft. And then this acetylcholine, next part is part number 3, will bind to the receptors on the postsynaptic dendritic spines. Okay, then this acetylcholine or neurotransmitter will binds to the receptors present on the dendritic spines of the postsynaptic neuron, of the postsynaptic neuron. And after it binds, what will happen is, I told you, now we come to the ligand gated channel. If you remember, now acetylcholine has bound as a ligand, and that will do what? Now this is a ligand gated sodium ion channel. Acetylcholine has bound ACH, ACH, and now it will cause Na plus 2 influx, that is depolarization. And if enough number of acetylcholines has been released and has bound, that will lead to spatial, you can say, summation, and that will lead to generation of gated potential. And if it can, you can say, increase the voltage from minus 70 to minus 55 millivolt, it has reached the graded potential, and then you can say the other voltage gated sodium ion channels can also open up. Now the voltage gated sodium ion channels can open. Okay, so now again, see, this neurotransmitter released is causing the action potential to be perpetuated in the next neuron. Okay, so that is causing the action potential to be perpetuated in the next neuron. Okay, so this is the entire event, if you can say, pertaining to the release of the neurotransmitter, okay, from the axon terminal slash telodendria slash synaptic end bulb slash varicosity. Now tell me, is there any doubt in today's lecture? Any doubt, any queries? So this is how the evolution takes care of the entire body, right, of the entire neuronal system or neuroscience, that the neurotransmitter released binds to the ligand gated channels and will cause the perpetuation of action potential in the postsynaptic neuron. That is why neurotransmitters are very, very essential, because otherwise there will be no neurotransmitter which will be binding to the, you can say, postsynaptic neuron, and if they are not binding, then current will not be propagating in the postsynaptic neuron. Without current propagation, no nerve impulse. Without nerve impulse, paralysis or death can also occur. Okay, so that is how amazing our body is. Okay then, thanks a lot. Let's meet you in the next class.